So good morning. Today's topic is enzymes. So what basically enzymes are? There are vast multitudes of chemical reactions that occur in our living organism. These reactions would proceed at extremely low velocities in the presence of catalyst. The common catalysts used in non-living systems are acids, alkalis, metals, which are not suitable for living organisms because of their toxicity and lack of specificity. The biological catalyst must be safe as well as specific. They should also be capable of adjusting their catalytic activities according to the ever-changing requirements of the organisms. All these properties are present in enzymes, which are biological catalysts used by living organisms. So we can define enzymes as a biological catalyst, and the proper definition goes as such that enzymes may be defined as protein catalysts that catalyzes chemical reaction in biological systems. They are protein catalysts, which means that biochemically they consist of what proteins are consist of. Proteins are basically polymers of L-alpha amino acids. So all the enzymes bearing ribozymes are of proteinous in nature, or they contain L-alpha amino acids as their monomeric units. We cannot say that all enzymes are protein because there have been some recent discoveries leading to the findings that certain RNA molecules, they act as catalyst, biocatalyst. So this definition is not entirely correct, as all enzymes are not proteins now, as some of the RNA molecules known as ribozymes have now been found to catalyze some biochemical reactions. Moving on to the next slide. In a simple process, there are substrate. In a simple reaction in metabolism, there are certain substrates which leads to the production of products, and enzymes work is to just catalyze the reaction, just to speed up the reaction. The next topic is enzyme specificity. The biological catalyst must be very specific, which is essential for normal functioning of the organism. If a single enzyme catalyzes a large number of reactions, it will be impossible to regulate the rates of a individual reaction. So the enzymes are specific in their function. What this slide says is that they are specific in their function. Every substrate, every reaction, particular set of reaction will have a separate enzyme. How we will see this? There are three kinds of specificity shown by enzymes. Enzymes are group specific, enzymes are substrate specific, and they also are stereo specific. What is group specificity? These enzymes are specific for chemical groups or bonds that they act upon, but not for the actual substrate. So there are certain type of bonds are there which are catalyzed by certain set of enzymes. So group specific or bond specific enzymes are frequently present in digestive system and hydrolyze the complex molecule of foodstuff. What well in digestive secretions and system, what our body does is that whatever macromolecule that we consume as diet, they are large in size and they have multiple types of bonds. So the basic aim of the digestion of food is to make those macromolecules into smaller molecules, just to hydrolyze them or to break them into smaller molecules, which could be easily absorbed by the intestine. In order to do that, there are certain sets of enzymes present in the secretions of digestive juices. And these enzymes, they break the macromolecule into smaller molecules known as monomeric units. For example, if we see pepsin, trypsin, and chymotrypsin, these three are hydrolyzing enzymes for the proteins, as they hydrolyze the peptide bond, but they would hydrolyze the peptide bond present in any protein. This gives an advantage to the organism in that A very large variety of dietary proteins can be digested by a small number of proteolytic enzymes. Other examples also include lipases for lipids and glycosidases for glucose and other carbohydrates, nucleases for nucleic acids. Some group specific enzymes have a slightly higher degree of specificity. For example, aminopeptidases and carboxypeptidases, they split off only the N and C terminal amino acid. What is N and C terminal? Basically, these are two ends of a polypeptide chain. Will N terminals represent basically the amino group of an amino acid. If you see in an amino acid, there are two certain groups. One is the carboxyl group, other is the amino group. So obviously, when those amino acids will join together to form a polypeptide, a linear long chain of amino acid, which is known as protein. So eventually, a protein will have two ends. One will be the N terminal, which contains the amino group, and C terminal, which contains the carboxyl group. So aminopeptidases and carboxypeptidases. So the name itself says that aminopeptidases will act upon the N terminal, and C terminal amino acids will be catalyzed by carboxypeptidases. And the next one, which is endopeptidases, since the name itself means endo, meaning inside the molecule. So they will hydrolyze the protein at somewhere in the mid of these two. By this example, we can see here. See, aminopeptidases, they are working towards the amino terminal, and carboxypeptidases, they are working towards the carboxy terminal of this long chain protein, which contains phenylalanine, glycine, cysteine, arginine, lysine, alanine, methionine, tyrosine, and valine. And see, the endopeptidase, it is working at the center of this molecule, the center of this chain. So these three are group specific enzyme amino acids, group specific enzymes, as they are functioning on similar particular sort of bond, which is the C-N bond or C triple bond N, which is known as peptide bond. These three enzymes are catalyzing the breakdown of uh, peptide bonds. Moving forward, the second sort of specific nature is of substrate specificity. Since most of the enzymes are specific not only for a chemical bond or a group, but are for substrate also. For example, glucokinase and fructokinase, these two are important enzymes present in glycolysis and are substrate specific enzyme. They transfer a phosphate group from ATP to one of the specific substrate, leading to the formation of glucose 6-phosphate or fructose 6-phosphate. We see here, these two are the steps of glycolysis. Here, glucose is converted to glucose 6-phosphate, meaning at sixth position, carbon position, a phosphate is added to the sixth carbon with the help of the enzyme which is catalyzing glucokinase. So glucose is the substrate and the name of the enzyme is glucokinase. Since kinase is here, kinase simply does what? They break the adenosine triphosphate into adenosine diphosphate, leading to release of one phosphate, which is added to the glucose at the sixth position. Similarly, fructokinase catalyzes a reaction in fructose by breaking down the ATP into ADP, the adenosine triphosphate into adenosine diphosphate, and then thus releasing one phosphate and then adding that one phosphate to the first carbon of the fructose. If you see here, the process, the end result is same. Even the ingredients are the same. The reaction, the catalyzing, which is happening is same. Only the substrate is different. Glucose is here, so kinase itself splits the ATP. Here fructose is here and fructokinase is here. It is catalyzing the reaction, leading to the formation of ATP. Breakdown of ATP is here. If we note system wise, we can see here ATP is breaking down into ADP. Triphosphate is converted into diphosphate. Similar in these two reactions. Similar release of phosphate is there, which has been added to the substrate, which is fructose and glucose. Only the difference is the numbering of carbon atom where the phosphate is added. These two reactions are steps of So we can say here that glucokinase and fructokinase, these two are catalyzing the same reaction, leading to the result, same result, but they are specific in their sense that they are using separate substrate, which is glucose and fructose. The next one is stereospecificity. Many biomolecules exhibit stereoisomerism, like carbohydrates and amino acids. Enzymes acting upon such substrates are usually specific for a particular stereomer. Mammalian enzymes acting on carbohydrates are generally specific for D isomer, and those acting on amino acids are generally specific for L isomers. Exceptions are racemases, which can interconvert the D and L isomers of a compound. The next topic is coenzyme and cofactor. Some enzymes require the presence of non-protein substance in their catalytic activity. If the non-protein substance is organic, it is known as a coenzyme. Basically, the non-protein part, which is known as prosthetic group, the non-protein part, which is known as the prosthetic group, contains two type of things. If it is organic, then it is known as coenzyme, and if it is inorganic, then it is known as cofactor. The coenzyme or cofactors may be integral part of the enzyme molecule, or it may be present only when they are required for a reaction. The protein portion of an enzyme that requires a coenzyme is known as an apoenzyme. The apoenzyme combines with coenzyme to form the holoenzyme, which is the catalytically active part of the enzyme. So apoenzyme plus coenzyme leads to the formation of holoenzyme, which is a complete enzyme. You can see here in the diagram, an apoenzyme which is in active state. When a coenzyme is added to it, it leads to the formation of holoenzyme, which is also known as your active enzyme. Active enzyme basically is an enzyme which is ready for its catalytic activity. The coenzyme generally contain vitamin of B complex family: thiamine, riboflavin, niacin, pantothenic acid, pyridoxine, folic acid, vitamin B12, etc. Some of these enzymes, some of these vitamins, basically lipoic acid and biotin, act as coenzyme by themselves. The others can be converted into coenzymes. Coenzymes are generally required in the group transfer reaction, like oxidation reduction reaction, transamination, and phosphorylation reaction. They can be divided into two groups according to the nature of the chemical group that they help to transfer. So coenzymes can be of two types, basically. Coenzymes involved in the transfer of hydrogen, and coenzyme involved in the transfer of groups other than hydrogen. See here the list of enzymes, coenzymes, which involve in transfer of hydrogen: flavin mononucleotide, flavin adenine dinucleotide, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, lipoic acid, and coenzyme Q. These all six of them we can see here in the electron transport chain in the mitochondria, where ATP is generated by chemoosmotic theory, which has been proved only through the transport of electron from one complex to another. Coenzymes which involve in the transfer of groups other than hydrogen include thiamine pyrophosphate or TPP and thiamine diphosphate, coenzyme A, pyridoxal phosphate, tetrahydrofolate, cobelamides, lipoic acid, biotin, and adenosine triphosphate, and similar nucleotides. The role of coenzyme: the enzyme acts upon in substrates and converts it into a product. The coenzyme may be regarded as a substrate or a second substrate in the group transfer reaction. The coenzyme participates in the reaction either as a donor or as an acceptor of the group that is being transferred. In the first reaction, glycerol is the main substrate and coenzyme ATP act as a second substrate. We can see here the reaction when glycerol is converted to glycerol 3-phosphate and then to dihydroxyacetone phosphate. These are reactions of glycolysis. And again, you can see here kinases are used. So ATP will be breaking down into adenosine triphosphate with the help of enzyme glycerol kinase. And in the second step, glycerol 3-phosphate dehydrogenase is where NAD and is converted to NADH plus phosphate plus proton, leads to the formation of DHAP or dihydroxyacetone phosphate. In the second reaction, glycerol 3-phosphate is the main substrate and the coenzyme NAD act as second substrate and accept the hydrogen atom. The chemical changes in the coenzyme is opposed to that in the substrate. Some coenzyme accept a chemical group from one substrate and donate it to other. Thus, they can act only as a carrier and regain their original form at the end of the reaction. So paradoxal phosphate is an example which act upon as a carrier of amino acids in a transamination reaction. We can see here the paradoxal phosphate, it is converting glutamate oxaloacetate transaminase GOT enzyme. It is converting aspartate and paradoxal phosphate into oxaloacetate and paradoxal phosphate. And this reaction is catalyzed by glutamate oxaloacetate transaminase. In the previous reaction, the transamination reaction catalyzed by glutamate oxaloacetate transaminase or GOT enzyme, paradoxal phosphate accept the amino acid group from aspartate and transfer it to alpha ketoglutarate. Aspartate is converted into oxaloacetate and alpha ketoglutarate into glutamate. The coenzyme goes back to its original form at the end of the reaction. Though paradoxal phosphate is a reactant in the transamination, the reaction is often shown as aspartate plus alpha ketoglutarate with the help of enzyme GOT and paradoxal PLP, leading to the formation of oxaloacetate, which is a 4-carbon compound, and glutamate, which is an amino acid. Sometimes the change in the coenzyme is more important than the change in substrate. For example, in aerobic glycolysis, glucose is converted into pyruvate and NAD is reduced in one of the reactions. So reduced NAD transferred its hydrogen atom to oxygen forming water and NAD plus is regenerated. In anaerobic reactions, NAD cannot be regenerated due to lack of oxygen. So therefore, a regressional reaction occurs in which pyruvate is reduced to lactate and NAD is oxidized to NAD. Here, regeneration of NAD is more important for continuation of glycolysis. You can see in the reaction, the glucose is converted to glyceraldehyde in glycolysis, which is mid step in glycolysis. Glyceraldehyde is a 3-carbon compound, and at the third position of carbon, phosphate is added. That converts into 1,3-bisphosphoglycerate, leading to the production of pyruvate when aerobic conditions are there, and if anaerobic conditions are there, then it will ultimately lead to the formation of lactate with the help of lactate dehydrogenase. And you can see here the NAD is converted to NADH plus proton and simultaneously NAD plus.